We're starting with the cardiovascular system, and there's three principal components to the cardiovascular system. And the first one we're moving into is the fluid blood. So we're taking a look at chapter 20 specifically, but we're going to have an introduction to the cardiovascular system. Our body has trillions of cells, so a large collection, and we need a way to just physically transport various chemicals and factors and just physically transport them around the body. And a general name for this is the circulatory system, but cardio, meaning the heart, vascular, the blood vessels, arteries, veins, capillaries. And again, it's the fluid blood that's truly in motion, delivering various factors. And what's carried and suspended in the blood, of course, there's various gases, and of course, especially oxygen, carbon dioxide. Hormones transfer through the body via our bloodstream. The nutrients that we absorb, waste chemicals produced and byproducts of metabolism from our cells, that's dumped into the bloodstream for ultimately removal from the body. And it's not only these tangible materials being circulated, but also temperature, warmth, as it transfers especially to the superficial areas of our body, our skin. We could transfer that heat from the deeper areas of our body and bring it closer to the surface. And the last two features, of course, we won't touch on too much in this course, but are erythrocytes, right, our red blood cells. That's one way that we help to regulate and maintain our body's pH. And we'll discuss various white blood cells. And again, those white blood cells, those leukocytes, giving us this immune function depending on their specific activity. So again, the blood is just the first component that we're going to approach for the cardiovascular system, and we'll follow along with chapter 20, seeing the tissue of blood. And chapter 21 is where we'll move into the pump for the system, the heart with its four chambers and valves. And chapter 22 is now looking at the vascular portion, truly the arteries and veins, and you can think of it literally just the system of tubes, the plumbing network essentially, that now propels and carries the blood. So the pathway for the circulation of blood are the vessels, the pump for the system is the heart, and truly the material that's in motion and delivering these substances is our fluid blood. And with this figure here, we just wanted to touch on the fact that it's not only various nutrients and gases, but again, the temperature dispersal. So with our warm blood, and we'll point out the temperature, but our blood is slightly warmer than our average body temperature. As blood flows to an area, it's also bringing that increased heat. And the example is right here. You can see this hand, and a person was typing on this modified keyboard for a period of 7.5 minutes. And over that time of exercise, that increased blood flow to their muscles and the working muscles themselves, giving off more heat. And you can see here in the forearm and in those fingers as well. So that exercise, both the working muscles and that increased blood flow, you can see the areas kind of heating up. So blood is a connective tissue. This goes pretty far back for us now to the beginning of the semester. But blood is one of those fluid connective tissues. And taking a blood sample, we're going to stick with these very generalized values here. Okay. Of course, there's variation from male to female among ethnicities. But with this really simplistic view here, taking a look at a blood sample, it's about 45% formed elements. And the term formed elements truly means the cellular components of blood. So red blood cells, white blood cells, the cellular portion is the formed elements, whereas the larger percentage, the plasma is to a high degree water with dissolved material. So a large amount of water with, of course, a dissolved component. We see that pH range for blood, and as we just mentioned earlier, slightly higher than our average body temperature, that 98.6 degrees, whereas blood, again, being slightly warmer. So as blood circulates, especially closer to the superficial areas of our skin, it's bringing that increased warmth. And about the amount of blood, here we're seeing, of course, based on the sex of a person, but truly more related to body size. So with typically bigger, muscles, more height, more body size, it's truly just the body size leading to the larger amount of blood that we see. So this difference between men and women. So if we start with this sample of blood again, it could be separated. And this is easy as using a centrifuge capillary tube to separate these two partitions. So starting with the plasma, and the isolated plasma does kind of have this yellow tint to it. Again, it's that larger percentage of the whole blood sample, and you see to a large degree water, with now the remaining material composed of various dissolved substances that are dissolved into that solution of the plasma. And again, these various components of the plasma are now in circulation and being delivered around the body. So with the plasma, we will touch on now these groups of proteins, albumins, globulins, fibrinogen proteins that are a major part of the plasma. And we'll touch on these three groups in more detail. Whereas the second component, there it is, the formed elements are truly the cellular makeup. And of the formed elements, nearly all of it, 99.9%, are what's commonly referred to as the red blood cells. So tackling the plasma protein component first, of these various plasma proteins, a large majority are made in our liver. And the liver has a lot of interaction with the cardiovascular system and the maintenance of our blood. And of course, we'll touch on that, and it'll come up again in the digestive system. So the albumins. So albumins as a category by itself, they form what's called these lipoprotein structures. So we have a protein structure that attaches onto a fatty lipid structure. And the importance of these lipoproteins is now, we just pointed out that our blood is a large percentage of water. And in order to transport fat, and steroids are a type of fat, in order to transport fat through this plasma watery based medium, we then combine them with these lipoproteins, and now we have the ability to transport nonpolar fat within this fluid basis of plasma, with this water medium of plasma. We have another transport protein, the globulin, and a principal structure they're involved in transporting are the antibodies. So the antibodies of our immune system. And the full term would be immunoglobulin and immunoglobulin. And we have that transport again, that binding, and transporting now this material again through our fluid-based plasma. And then a third category of proteins, we have what's called the inactive state. So fibrinogen truly alone is inactive, but when activated and converted to the active state, the functional state, fibrin, this is now the network of fibers that are now the ultimately making this framework of now insoluble fibers. And this is one major portion of a blood clot. So when there's an injury, we have this active state of fibrin, and we now make this network of fibers, and that's one stage in forming the blood clot, and of course promoting the healing process. And ultimately, after injury, we can now stop the bleeding and start the repair process. And here's just a figure illustrating this here. So we can see the fibrin strands that form, so making this network here. But we do also notice that there's various formed elements that also are now embedded in this fibrin network as well. So moving now to the formed elements, again, the cellular features. But it's also important to note that not every formed element truly is a cell. Some of them are simply broken fragments, broken pieces of what used to be a cell. So the three principal groups are now using the scientific terms. Erythro meaning red. So we have the mature red cells, the erythrocytes, and their purpose is principally transporting the gases, oxygen, carbon dioxide. Leuco meaning white, so the mature white blood cells, the leukocytes, and they have various immune functions in promoting the healing process. And then now, truly, is the third group here. This is where the fragments come into play. So platelets are truly just broken pieces of what used to be a larger cell. And we notice here again this description for blood clotting. So it's both the fibrin plasma protein, and the platelet formed elements. Both of these are involved in making a proper blood clot and, of course, preventing further blood loss after injury. So if we start here with this figure, introduction to the chapter, if we have a whole blood sample, and again, using a centrifuge, we could separate it. And now you start to see the broader range of variation. So the percentage of plasma versus formed elements. In the plasma, as we just mentioned, of course, large percentage of water. And then we've touched on these three categories of plasma proteins. So each of those, the brief description as we touched on. Taking a look at the cellular components of formed elements, nearly all of which, and now we're using the term erythrocytes. A much smaller percentage, of course, for the various types of leukocytes, and you notice here the 5 principal groups. And then now those broken fragments, what we call the platelets. So we'll move through the various types of formed elements. So these blood cells, these connective tissue cells, they have their formation, their creation. This term hemopoiesis is one term for the creation of blood cells, the synthesis of blood cells. And we notice in our fetal stages before birth, there's multiple locations that are important for rapidly producing blood cells within the developing fetus. However, as we get closer to birth, so close to birth and then after birth for the duration of our life, we see that the major sites after we're born are the red bone marrow of the various bones of our body. But we also previously studied yellow bone marrow, and that's a secondary site. Okay, when there's a larger need to produce more blood cells, you can imagine after injury, after some large amount of blood loss, the yellow bone marrow could be used as a secondary site for hemopoiesis. And the cell involved in producing and creating all types of formed elements. You see the term here, hemocytoblast. So just think about that ending there, that root term, the blast. So that young cell that could differentiate into forming all the various types of formed elements. So a nice figure to represent this here from our textbook. And starting from that hemocytoblast, and we're just concerned with the ultimate outcome. So just all the way down to the bottom of the chart, Hemocytoblasts, of course, can differentiate into our erythrocytes. You notice here there's this step with this very, very large cell that then fragments to make our platelets. And we have the various classes of leukocytes. Also, this figure here, this is showing now, you notice the weeks of development. So from our embryonic stage and then on to our fetal stages up until birth. And before birth, we see there's multiple organs involved in producing blood cells. However, as we get closer and closer to birth, it really now becomes the responsibility of the bone marrow, and chiefly the red bone marrow, and after birth to make this source of blood cells. So we want to start with the mature erythrocytes. And as we already pointed out now, the large majority, nearly all of our formed elements, hence the ratio. And we have this figure, this measurement called hematocrit. And this is truly looking at, just as it describes, the percentage, the concentration of erythrocytes in that particular amount of blood. So per some amount, some particular sample of blood, what percentage of that is made of the erythrocytes. And that's really where we got that estimate of the formed elements. Earlier, we gave the very simple number of 45% of the blood sample being the formed elements. And again, that's just a very rough estimate. There's a lot of variation. The measurement is truly taken directly from the hematocrit concentration, but since nearly all of the formed elements, 99.9%, are erythrocytes, we could then use that hematocrit level to estimate the total concentration of formed elements. Just some interesting traits about the erythrocytes. They lack multiple organelles. However, lacking these various organelles here, especially ones that consume oxygen, make these cells ideal for the now just transport and delivery of oxygen. Within a single erythrocyte is filled now with this protein. This structure, hemoglobin, functions as both a protein and a pigment. The pigment, of course, allowing for the color, and the color that we see of our blood comes from hemoglobin, so that state when it's bound to oxygen. But looking at its protein structure, this is now the particular protein that actually binds and transports the various gases of our bloodstream. So that direct interaction with the hemoglobin. So as an example, oxygen, areas of our body that have a high concentration of oxygen, and this of course would be the lungs. As we inhale and we're drawing that oxygen from the environment into our lungs, the blood circulating through our lungs will now bind oxygen to the hemoglobin. And now when this oxygen-rich blood, this oxygen hemoglobin-bound blood, circulates to other areas of the body, so you can imagine here in your working muscles, for example, your working skeletal muscles that are consuming oxygen and demanding more, the hemoglobin releases that oxygen and it's allowed to diffuse into those working organs. So truly, erythrocytes just cycle through circulating through the lungs, loading up with oxygen, and then delivering that oxygen to all our various organs and tissues. And again, a single erythrocyte has a huge oxygen capacity. So with millions of these hemoglobin proteins and a single hemoglobin molecule, again, having this capacity to transport 4 oxygen molecules, so giving us this tremendous capacity to transport oxygen through our bloodstream. Within a single heme group, if we take a closer look here, you can see the center of that heme group involves iron, and that's ultimately the direct interaction between oxygen and iron, making that binding into the hemoglobin. And lastly, it's not only just oxygen that binds to the hemoglobin, but carbon dioxide as well. The benefit, of course, is oxygen is being bound in the lungs and released at our organs, whereas carbon dioxide is doing the reverse trip. Our blood and our hemoglobin binds carbon dioxide at our organs and then releases that at our lungs, and we're able to exhale and remove that carbon dioxide as we exhale. So these gases are transported by the erythrocytes, but the gases are also dissolved directly into the fluid plasma as well. So the final trait about the erythrocyte is, of course, our blood typing. So when it comes to our what's referred to as the ABO grouping, type A, type B, whatnot, that's now a trait of the erythrocyte. So the diagram here shows these 4 different blood typing groups here, generalized. So going through these various groups, it comes down to the particular type of surface antigen, this marker that's shown and exposed on the cell membrane of erythrocytes. So what's known as type A blood, these erythrocytes are marked here with the surface antigen A, so represented here by this red circle. Or type B blood has the surface antigen B, so represented here by this blue triangle. AB type blood has both of the surface antigens, both A and B. And the type O grouping lacks either one of those. So we have the A grouping, the B grouping, having both of these. That makes this type AB. And lacking these two leads to the type O. So now next we want to discuss the plasma itself. So keep in mind the erythrocytes are part of the formed elements, the cells, the plasma, the fluid. So matched with each of these types, the plasma of a type A person, their body now is developing anti-B antibodies. The blood type B, in their plasma, they're producing and continually generating anti-A antibodies. Type AB person, they're not producing either one of those anti-A or anti-B antibodies. And you see here the type O grouping in their body and in their plasma, they have the production of producing both anti-A and anti-B antibodies. So what these antibodies do is, if these antibodies will stick with anti-B, for example, if an anti-B antibody comes into contact with a B type of erythrocyte, it'll have this immune response, the process of antibody, to actually kill off this B type of erythrocyte. So a person with type A blood, their immune system's constantly producing these anti-B antibodies. And again, if this B type cell was introduced into the A type person's body, the anti-B antibodies would target that and attack and destroy that cell. And that's when blood typing comes to be very, very important. There's certain types that could be safely donated, and there's other types that would be, of course, very harmful if donated, even fatal, of course. So one last, of course, issue we want to take a look at before we get into blood typing is the Rh factor. And either the Rh factor is present, and that's indicated as positive, or if the Rh factor is missing, then we would say that person is Rh negative. So to illustrate that a little better, let's change colors. So if the Rh factor is present, That now leads to the distinguishing trait of being a positive type. So we can modify our figure here. We'll say the RH is just a green square. So now this is a positive because they have both the A antigen, and now we have this green square representing the RH. So it's now a positive type blood. Or if we look over here, just using this textbook image, there's the A, there's the B, but since we didn't draw an RH, this would be truly AB negative. If we wanted to make it to a positive type, let's just draw in our little green box. So now the RH is present, and we could call it AB positive, and so on. And the positive negative is also involved in proper blood transplantation. So a simple way to learn the donation and recipient interactions and what's safe, we have a simple diagram we could draw here. We have the type O blood, and we'll draw an arrow coming across to AB. And then we'll draw the other two types. Here's A, and here's B. And for this diagram and for this proper type of transplantation, the arrows are always going to go towards the right. So if we make this little sketch here, just drawing the arrows just only going to the right. So what this is showing us is type O could be donated safely to all three other types. Type A could be donated to AB. Type B could be donated to AB. And then the last thing we want to draw in is each type could be donated to itself. So we'll just draw an arrow pointing back to each of the same letters. A could be donated to other A types. And then AB can only be donated to other AB types. So in terms of the ABO grouping, this is what we see here. But now there's still this issue of RH factor. So the only theoretical possibilities is donating positive blood to another positive person, a positive person donating blood to a negative person, negative being donated to another negative RH type, or negative being donated to positive. So these are the four possible donation pathways and possibilities. However, one of them is not safe, and we just want to highlight that one, and we'll cross it out. So the one that would have to be avoided is seen right here. So we would not want to donate positive blood to a person that has RH negative. So we'll just highlight that one and just put a big cross on it. So if we look at these two different now little charts, we see in terms of the lettering, the ABO groupings of which can be donated to which. And now with the positive negative interactions, as long as it fits in with these three types for the RH donation, and it follows this diagram here, that would be a safe donation. So for example, you notice how both negative types, RH negative, could be donated to other negative RHs. RH negative can be donated to RH positive. So simply put, it's safe to donate a negative type to either type of blood. And here we see O could be donated to all different types of letter grouping. So just following that logic, O negative, you may have heard, O negative is the universal donor, and O negative type blood can be safely donated to anyone. Just following the diagram, O can be donated to any of the letter grouping, and negative can be donated to any type of RH grouping. So the last word truly comes from what makes it unsafe for a donation. And again, to get some of the writing off of the screen, it always comes down to the type of antibodies that the recipient, the person receiving the blood, what type of antibodies do they have? If their antibodies would fight off that incoming erythrocyte, so right here I'm drawing this diagram of donating B type to an A type person. The A type recipient would reject it because their anti-B antibodies would reject and attack that incoming B type of erythrocyte. So it's the recipient's antibodies that's the key in seeing if that's a safe donation. So the final bit here, just an interesting trait, taking a look at the distinct shape of, this here normal erythrocytes, described as a biconcave disc. And this particular shape is extremely helpful when it comes to being just a very tight fit, a very tight squeeze, passing, this here stacks of erythrocytes through a small capillary. And a genetic disorder, genetic disease, this mutation known as a sickle cell trait, that abnormal shape, of course, that could compromise oxygen absorption and transfer. It could lead to literally clogging. It's called a sickle cell crisis, clogging in the erythrocytes. And of course, compromised oxygen transport. So now moving on to the leukocytes. And again, that's the term we want to use now, the leukocytes, that's a scientific term. So these cells typically have some type of immune function, so responding to infection, and of course, ultimately promoting healing and recovery. They have a relatively short lifespan compared to erythrocytes, which erythrocytes can last and develop, persist for a few months. These are, of course, a much shorter lifespan. Again, when we're healthy, these are in such a small percentage relative to the very, very common erythrocytes. And again, all blood cells, especially after birth, of course, have their development first in the bone marrow. So even though very few of these leukocytes are found within our blood, the large percentage of these cells are found in various other organs. So outside of the cardiovascular system, especially our lymphatic organs. And just looking at their relative abundance, I always like this little mnemonic here. Just never let monkeys eat bananas. Of the leukocytes, the neutrophils are the most common. And in descending order, we see here the basophils are the least common type of leukocyte. So with these 5 different categories here, the leukocytes are then divided into 2 groups. And this was an easy way that I remember this. We have what's called the granular leukocytes. And again, they all end that term. They're the phils. So taking a look at their structure, what it means to be granulated, if we make a little sketch of a leukocyte cell, and I'll go ahead and I'll just draw a eosinophil to be accurate. So we can shade in the nucleus here. And this is one single nucleus. It's just a bilobed single nucleus. What's meant by granular is when you see the cytoplasm, it has this granulated or kind of speckled appearance. So I'm just trying to draw on a few little dots here. So that speckled, granulated appearance to the cytoplasm, that's what all the various phils have in common: neutrophils, eosinophils, basophils. So this first category, this subdivision of leukocytes, the granular leukocytes. So of which the neutrophils, they have this more specific function in responding to bacteria. So an infection of bacteria, we have the neutrophils to now target that infection and trigger that immune response. Eosinophils, or you notice here an alternate name for them. Eosinophils, commonly involved, of course, in responding to allergens, or of course in other extreme cases, as well as parasites as well. So again, foreign material, foreign compounds, and we start that immune response. Whereas you see here, basophils, they're now involved in the inflammatory response. Inflammation, increased blood flow, that blood's bringing that warmer temperature. So increased blood flow, increased temperature. That, of course, is one way to fight off infection. So that stimulation, that stimulating the inflammatory response, thus a generalized way to respond to infection. And of course, it's delivering more blood cells to that area as well. So these first three examples, the granular leukocytes, whereas the other category, the remaining two, the agranular leukocytes. So again, making a little sketch, and we'll make a sketch of a lymphocyte. So each type of leukocyte does have a distinct appearance to its cytology. But again, what's meant by agranular, in this case, the cytoplasm is free of granules. So this time we won't draw in any little speckles. So the lymphocytes, again, this is just a broad category of cells. And just some examples of various types of lymphocytes. There's T cell lymphocytes, B cell lymphocytes, as well as others. So now, just to keep it more generalized, you notice the term lymphocytes. These cells are in very high concentrations, again, in our lymphatic system, our lymphatic organs. And you can see where that name comes from. The T cells, a category and a subdivision all on its own. These are the types of cells that, of course, can detect and respond to infection and really trigger that cascade of immune responses. B cells in particular, this is source of now producing antibodies. And now the antibodies that we develop can be specific to various infections, various pathogens. So we have diversity in producing various types of antibodies. And again, the antibody itself, a part of our immune response. Lastly, the monocytes. These have a more just generalized immune function, circulating around in our tissues, passing into our various structures as well. So these cells and all white blood cells have the ability to exit the cardiovascular system, migrate into various tissues, and in this case, this is where we have the term, the name change. Monocytes are located in the cardiovascular system. So monocytes are located in the blood. And once this particular type of leukocyte exits the cardiovascular system, when it's embedded in the tissue, these are known as macrophages. But again, they have their generalized immune function, trying to take in and break down foreign material. So we'll finish this section on leukocytes just with the image here. So again, all of these leukocytes have the ability and the need to exit completely out of the cardiovascular system and go into that infected or injured tissue, and of course, do their specific immune response, their specific function. So the final part for the formed elements, the red blood cells, the leukocytes, and now the platelets. And I just want to point out here a former and now incorrect term. We'll just go ahead and cross out this word, thrombocyte. The meaning would be, of course, a cyte's a mature cell, but we now know platelets are not cells. We'll cross that out. Platelets are just simply fragments of what used to be this large cell. So that original cell known as a megakaryocyte, a very large cell, And as a megakaryocyte breaks apart, the small little fragments of cell membrane and just the various amounts of cytoplasm, those ruptured components of the megakaryocyte, now become platelets. And as we touched on earlier, it's both the activated state of fibrinogen when it's converted to fibrin. So fibrin, of course, coming from our plasma, fibrins from the plasma. And now we have the platelets as a part of our formed elements. So it's both the fibrin and the platelets functioning together. Let's just spell this out. So the fibrin and the platelets, I think I messed up there. Yeah, the fibrin and the platelets, trying to draw that nicely. Together, both of these are now involved in blood clotting. So we need two halves there.